This is one of a series of good answers presentations, offering evidence to refute skeptical challenges to the Bible's accuracy. Critics of Christianity have found many ways to attack the Bible. One of the most effective has been to attack it at the most basic level. They challenge the existence of God. If God doesn't exist, then the Bible is only a collection of ancient writings, having no more value than any others. It can be put back on the shelf to gather dust, along with the Egyptian Book of the Dead or Aristotle's Poetics. Perhaps of value to some, but only if one has an antiquarian interest. The best tool men have ever devised for denying God's existence is Darwin's theory of evolution. Some years ago, Richard Dawkins, one of the most famous evolutionists alive today, was being interviewed on television. The interviewer asked him what benefit he found personally from Darwin's theory. Dawkins replied, "Darwin made it possible to be an intellectually fulfilled atheist." His reasoning was that before Darwin published his famous Origin of the Species in 1859, anyone who wanted to deny God had a problem explaining how everything we see came to be. Darwin's theory provided an explanation that one could find satisfying. Now, of course, a Christian can believe in the theory of evolution. Men do. But a serious look at the theory shows that its reason for existence is to make God unnecessary. As Philip Johnson once so tellingly remarked, an evolutionist will have no problem with saying that God exists, so long as just existing is all he does. Although many Christians do incorporate evolution into their understanding of the world, I would argue that it compromises our understanding of the early chapters of Genesis, and that, over time, it has a corrosive effect on our respect for Scripture. What is more, a close study of the theory reveals that it has serious flaws at almost every point. One of them concerns the origin of life. Can scientists make life? They certainly want to do so, and news reports show how optimistic they are. We regularly see news releases claiming, like these, that they are on the verge of success. But when we look beyond the headlines, the actual situation is less encouraging. The article in The Verge, dated March of 2014, is typically exaggerated. Despite the claim of almost making life from scratch, the article's text describes how a crew of graduate students painstakingly cut and pasted existing DNA and inserted it into living yeast cells. That's hardly working from scratch. But there's no shortage of enthusiasm. It seems that there are troops of scientists out there who want to be able to shout, like Gene Wilder in the film Young Frankenstein, "It's alive!" The Dr. Frankenstein approach, sewing together body parts and zapping them with electricity, dramatic as it is certainly never going to work. We need only think of the fact that if a heart attack victim, dead only a few hours, is brought into the lab and given a heart transplant, he should be a perfect candidate to be revived. All his organs are healthy and in place. Surely only some stimulation will be needed to bring him back to life. But that never succeeds. So let's be less adventuresome. Let's just see if we can make a single living cell. How hard can it be? Kids in school make models of the cell all the time, including labels for its parts like nucleus, Golgi apparatus, ribosomes, cytoplasm, and so on. One of the nice things about a project like this one is that, after it's graded, you can eat the whole thing. Certainly we see ample confidence among scientists. According to Andrei Finkelstein of the Russian Academy of Sciences, The genesis of life is as inevitable as the formation of atoms. There is life on other planets, and we will find it in 20 years. That's from June 2011. So, what do we need to build a cell? We would have to start, certainly, with what we find in nature, the 92 naturally occurring elements. Combining them in the correct order and arrangement, we can come up with amino acids. To make a cell, we'll need about 20 kinds of amino acids. These, in turn, are the building blocks for proteins. No one knows exactly how many kinds of proteins exist, but there are at least 30,000 kinds. Of the 20 types of amino acids, we will need a few hundred to make a simple protein. 5 of this kind, 12 of another, 3 of a third, and so on. And then we will need many different kinds of proteins to assemble a cell. But once we have all the different proteins we need in the right combinations, we finally have a cell. And it's alive. The search for life has been going on for a long time, and perhaps the most famous experiment is that of Stanley Miller and Harold Urey, 2 graduate students in the 1950s. They set up an environment to duplicate the conditions on Earth in the very remote past, over 1 billion years ago, as they believed. They assumed that the atmosphere would have consisted of methane, water vapor, ammonia, and hydrogen. To this they added a spark, thinking that the first life might have been created in an electrical storm. They ran the experiment for a while, then drew off the result, and actually found that amino acids had formed. The media went wild at their announcement. We've seen how the building blocks of life could have formed naturally. Despite all the excitement their work produced, the experiment had several serious flaws. First, they did not include oxygen in their ancient atmosphere, and for good reason. They understood that oxygen will degrade amino acids as soon as they are formed. After all, it is a corrosive agent, as any piece of rusty iron will tell us. But we now know from geology that oxygen has always been present in the air. Among the oldest rock layers we have found, there are traces of iron oxide. A second flaw was that spark they used. Only by drawing off the amino acids as soon as they were formed could they keep them intact, because the spark that created them would in turn have destroyed them. They could manage this in the lab, but in an ancient electrical storm, any amino acids produced would soon have been destroyed. Third, and the most serious of the flaws, is the fact that their experiment produced both right and left-handed amino acids. Each amino acid exists in 2 possible forms, you see, mirror images of one another, like our 2 hands. That's why they are called right or left-handed. The issue here is that all living cells contain only left-handed amino acids. The presence of even one right-handed acid would ruin the process. So what they produced could never have been used to make proteins, much less a cell. Scientists have tried again and again, by the way, to tweak the Miller-Urey experiment to get just left-handed amino acids. So far, they have failed. But despite all this, many biology textbooks still list the Miller-Urey experiment as a huge breakthrough on the way to making life. Any fair assessment, however, shows that it has been utterly discredited. But let's say that we can get around problems like right and left-handed amino acids. Let's forge ahead building a protein. As mentioned earlier, we'll need about 500 amino acids, distributed among the 20 types that are there available. Now there's the task of assembling these amino acids in exactly the right order and 3D arrangement that our target protein requires. We'll need a set of instructions, and here everyone knows where to turn: to DNA. This is the amazingly complex double spiral molecule that contains all the information needed to make a whole living person. It certainly contains instructions on how to make the protein we want. That it does. Ever bought something labeled "Some assembly required"? Then you know how important it is to read the instructions. The instruction for making our target protein is in the DNA, but it has to be read. For that, we need another very special molecule called messenger RNA. It works like this. The actual information in the DNA is in the individual rungs of the spiral staircase that the molecule resembles. To read that information, the DNA molecule has to be pulled apart so that the messenger RNA, the blue strand in this diagram, can snuggle up to the information-bearing stretch and copy what it finds there. Then the RNA heads off to another part of the cell where construction takes place. Like a coded tape, the RNA runs through one after another of a series of helper proteins, some 75 of them, that will use the information to pull together raw materials and build various portions of the protein that we want. And if all this coordinated activity works as intended, we will finally get our single protein molecule. Uh-oh. Do you see a problem here? We're trying to make a protein, right? And to do that, we need about 75 proteins. But if we're back at the very beginning of history, with just a mishmash of amino acids around, how are we going to assemble that first protein? Our problem is rather like this. Suppose we want to build an electric drill. We look on the internet and find a nice cutaway drawing that we can use as our model. It comes with instructions, and one of the instructions is, drill holes here. But we don't have the drill yet. Our plan to build a protein is just like that. We need proteins to build a protein. And you may have noticed, too, that the whole process of building proteins takes place inside a living cell. Well, let's not give up yet. We can probably find a way around these roadblocks. But one thing is sure. We need our instructions, and that means we have to have DNA before we can start to do anything. And DNA is pretty amazing stuff. We've seen that the information it contains is actually on the rungs of the ladder of that spiral. The bits of information themselves are like the letters of our alphabet. Biologists call them C, A, T, and G for short. So they constitute a 4-letter alphabet, and just as our alphabet is capable of forming countless different words, so is this one. It is effectively a language, much like a computer programming language, and like Fortran, C++, or Visual Basic, it can express very detailed information. A curious person might ask, where did DNA come from? That's a very good question. Evolutionists have offered only 2 possible answers to it. First, it may have arisen by chance. Don't count on that answer. Saying it came from chance is like saying that we could take a huge bag full of Scrabble tiles like these, shake it up and dump it out, and find that we have a complete set of the Harry Potter books. The chances of that happening are about as good as the chance that the DNA molecule, with its immense fund of detailed complex information and instructions, happened by chance. We'd be wise to note what Bill Gates said once. Human DNA is like a computer program, but far, far more advanced than any we've ever created. No, DNA didn't happen by chance. The other answer we hear is that DNA simply evolved from some much simpler molecule. There's trouble there, though. Evolution is defined as gradual change over generations. So that simpler molecule that was going to grow up to become DNA would have to reproduce generation after generation with tiny changes in each generation. Those changes occur in the DNA and are passed on to the next generation. So, to reproduce, an entity needs DNA. Without it, there can't even be a next generation. And yet, DNA couldn't have evolved from something simpler unless that something already had DNA. Another one of those circular logical traps, and no way out of it. Well, if chance can't have produced DNA, and it can't have evolved, then how did it arise? The only answer the secular scientist has for that one is time. Somehow they believe that, given enough time, just about anything can happen, no matter how impossible it may seem. They bring up the old monkeys and typewriters analogy. Have you heard this? It was Julian Huxley who popularized the idea back in Darwin's day. Put a bunch of monkeys in a room, each one at a typewriter, and let them bang away at the keys. Sooner or later, one of them will come up with the complete works of Shakespeare. Impossible? No, said Huxley. It may take a few million years, but sooner or later, one of those monkeys will get lucky. This argument actually sounds pretty good. A good friend of mine, both intelligent and a Christian, once repeated it to me. Sounds good, all right. Until you do the math. Here we go. Let's make it easier and not ask for all of Shakespeare. Let's just look for the first sentence of Hamlet's famous soliloquy. To be or not to be, that is the question. Just 10 words. And let's get plenty of monkeys. In fact, let's cover the earth with monkeys, each with his own typewriter, and have them bang away at 10 letters a second. That's pretty good for a human typist. How long will it take for just one of those little guys, clattering away, to come up with those 10 words? How long? 30 billion years. Believe it or not, 30 billion years. Walter Remine, in his book The Biotic Message, lays out the math in detail. His example is slightly different. He asks for a 100-word sentence from Hamlet. But he will let us use as many monkeys as there are protons in the observable universe. 30 billion years. That's twice as old as evolutionists say the universe itself is. Now, let's not lose track of our project, though. We are trying to build one protein on our way to making a single cell. How complicated is that protein? Let me show you a snippet from a video about one. This video is about processes that go on inside a living cell. It was produced by one of the most respected universities in the country. This part shows a computer-generated sequence about what's called a motor protein.
Membrane-bound vesicles travel to and from the plasma membrane. The directional movement of these cargo vesicles is due to a family of motor proteins linking vesicles and microtubules. Membrane-bound organelles like mitochondria are loosely trapped by the cytoskeleton.
We are way down deep inside a living cell here. From the nucleus, hundreds of strands called microtubules run out in all directions. We see a motor protein walking along one of those microtubules, pulling its cargo, that big blue object. You see, inside a living cell there is constant activity. Small parts of the cell, called organelles, are constantly being built and moved about, and as they wear out, they are disassembled and recycled into new organelles. You might say that the motor protein is the UPS of the cell. Its job is to locate an organelle, grab hold of it, find a microtubule, and pull the organelle to its proper destination, and then release it and head back for another pickup. It has to know how to do all of this. And yet, what is it? It's just a protein molecule. Proteins are not even alive. That is, they don't fit the definition of life. Only a complete cell can be said to be alive. And yet, somehow, it does its very demanding job. Here's another example, but this time it's not a single protein, but a combination of about 40 of them. It's the flagellar motor that some bacteria use to propel themselves around.
I remember the first time I looked in a biochemistry textbook and I saw a drawing of something called the bacterial flagellum with all of its parts in all of its glory. It had a propeller and the hook region and the drive shaft and the motor and so on. I looked at that and I said, that's an outboard motor. That's designed, that's no chance assemblage of parts.
Behe's reaction was not surprising, for the molecular motors that drive bacteria through liquid each depend upon a system of intricately arranged mechanical parts. These parts come into focus when portions of a cell are magnified 50,000 times. Biochemists have used electron micrographs like this one to identify the parts and three-dimensional structure of the flagellar motor. In the process, they have revealed a marvel of engineering on a miniaturized scale.
Howard Berg at Harvard has labeled it the most efficient machine in the universe. These machines, some of them are running at 100,000 RPMs and are hardwired into a signal transduction or sensory mechanism so that it's getting feedback from the environment.
And even though they're spinning that fast, they can stop on a dime. It only takes a quarter turn for them to stop and shift directions and start spinning 100,000 RPM in the other direction.
And just like outboard motors on motorboats, it has a large number of parts which are necessary for the motor to work.
The bacterial flagellum, 2 gears, forward and reverse, water-cooled, proton motive force. It has a stator, it has a rotor, it has a U-joint, it has a drive shaft, it has a propeller, and they function as these parts of machines. It's not convenient that we give them these names. That's truly their function.
Like an outboard motor, the flagellar motor that drives these bacteria is a complex assembly of parts. Take away one of the parts, and the motor won't function, just as when you pull a spark plug from an outboard motor. Molecular devices like these are said to be irreducibly complex, because they have to have all of their parts all in the right relationships in order to function at all. They could not have evolved from something simpler, because, lacking at least one part, they would have no use or value at all. They have to have been designed. Well then, as we look at a single living cell, we don't see, as Darwin did, something simple like a blob of jello. We see an immensely complicated, smoothly functioning machine with literally hundreds of interacting parts. And the truth is that we don't know how to build any one of those parts. From amino acids to proteins to molecular motors, we see a wonder in miniature. So how complicated is a cell? Much more so than the diagrams we can see in biology textbooks. And the deeper we are able to look into this subminiature world, the more complicated it gets. Life is the great mystery. Even now, in the 21st century, after generations of effort by thousands of our best and brightest people, using the most sophisticated tools we possess, it still eludes us. And yet, we look back to Genesis 2:7 and read, "The Lord God formed the man from the dust of the ground, and breathed into his nostrils the breath of life, and the man became a living being." Our people in the white lab coats have a long way to go. If you have questions or comments about this lecture, or any of the others in the Good Answers series, you may direct them to me, Dr. Jerry C. Four, at yahoo.com.